الله الرحمن الرحيم. شكرا جميعا ثلاثتكم طلع عن نفسي يعني عندك تعليق مستفيد يسعدنا أن يسعدنا أن نرحب بكم في هذه الورشة اليوم، ويشرفنا حضوركم كصناع قرار ونخبة من العلماء، ونثمن عاليا ونقدر مجهوداتكم لدعم العلم والتنمية، ونتمنى لكم يوما موفقا بإذن الله. يسعدني الآن أنه أدعو الدكتور البروفيسور نجلاء العبد الله، أستاذ الوراثة في جامعة القاهرة، واللي هي المنظم الرئيسي لهذا المؤتمر. تفضل لإلقاء الكلمة. عشان تسجيل بكل.
كيف جميعا، معلش هقول كلمة بالإنجليزي كنت عاملة حسابي إن أجانب. Good morning, everybody, distinguished guests, esteemed colleagues, respected scientists and researchers, value stakeholders and partners, ladies and gentlemen. Good morning and welcome. It's my great pleasure and honor to welcome you all to the opening of our workshop entitled Fundamental Application of Gene Editing and its Replication. The workshop brings together an outstanding group of national, regional, and international experts in the field of gene editing, biotechnology, biosafety, regulation, communication, and regulation innovation. It reflects strong collaboration among leading industry institutes, including the Faculty of Agriculture Research Center, AGIRI, and of course the International Center for Agriculture Research in Dry Land, ICARDA, and the International Institute of Tropical Agriculture, the Academy of Scientific Research, AAIT, and of course the Academy of Science and Scientific Research, of course, is a main partner in this event. At the outset, I would like to extend my sincere welcome and appreciate to our distinguished guest who's greatly honor this event. A very special welcome to Professor Dr. Ahmed Dab, acting Vice President of Academy of Science, Scientific Research and Technology, for the contribution, support, and scientific research, innovation, and national capacity building. Professor Dr. Adil Abdulazim, and we talk on behalf of him, Dr. Mujahid Ammar, Director or Head of Institute of Crop Institute. Okay, sorry, welcome. And he's going to talk on behalf of him. And also, I would like to thank them for their leadership in the advancing agriculture research and simple innovation-driven solution for sustainable agriculture. Also from the ICARDA, we have Dr. Michael Baum, Senior Scientist Advisor for Genetic Innovation at ICARDA, whose remarkable contribution to crop improvement, climate resilience, and international agriculture research continue to support agriculture development across dry region. Of course, we would like to welcome Dr. Lina. She came especially for this event from Kenya, and she has a good contribution with us in the workshop and also in the different kind of project. I also like to warm welcome our distinguished guest in the international expert, Dr. Lina from IITA, whose pioneer work in gene editing and biotechnology application in Africa has made significant contribution to crop improvement and food security. Your participant added tremendous scientific value to this workshop, and we are truly grateful for your presence. Also, I extend my thanks to Professor Piet van der Meer from Ghent University from Belgium, and Jean Komen and Lino van der Leen from Green Orange Global. He is going to talk in online. Also Sheila Obim. Sheila, she's the head of Alliance of Science, and we already have a lot of contribution and workshop together. Ladies and gentlemen, we are meeting together at a time when agriculture worldwide is facing major challenges: climate change, water scarcity, soil degradation, salinity, extreme temperature, and then the spread of pests, increasing spread of pests and disease, all of which place enormous pressure on food security and agriculture sustainability. This challenge requires solutions that are faster and more precise and more sustainable than conventional approach alone. This is where gene editing technology, especially CRISPR-Cas system, have become transformative. Gene editing have revolutionized plant science by a long, precise improvement of traits such as drought tolerance, salinity resistance, heat resistance, disease, and so on, different kind of effort. At the time, while scientists of CRISPR is precise, the science of CRISPR is precise, its path to participate application and market adopt is often complex. Through the workshop is distinguished to facilitate, this workshop is distinguished to facilitate dialogue on how we can create science-based and robust regulation framework that foster innovation while maintaining the highest standard of safety, policy, and ethics. While we focus navigating the complexity of biosafety development, share best participant and promoting responsible innovation with global accessibility. Our goal is to move from theoretical discussion to actionable steps. So as we know that gene editing is a new technology that gave a lot of promise for scientists and for different, for the improving of the different country income and solve a lot of problems to the farmers. We are pleased today's program reflect this version overcoming gene editing fundamental agriculture application in Africa, global and regional regulation, regulatory experience, science communication, strategy, and final panel discussion with stakeholders and policymakers. As you see here, we invited people from the private sectors, from different stakeholders, scientists, and international organizations, so we can get by conclusion by the end, and we can raise it for the stakeholders and who can, the decision maker who can take the decision to approve the law for gene editing in Egypt compared to the other African country who speed us and already have the law, and they are already implementing this new technology. Okay, I'll go to the end. Because the future of agriculture will not be built by traditional alone, it will be built by scientists, innovation, and responsible leadership. I would like to thank you all for. I would like to thank you all for your attendance and for participating in this important event. And I would like to welcome our first speaker, Dr. Michael Baum. Thank you.
Good morning, everyone in the room and online. It's a great pleasure to be back in Egypt, and I would like to welcome you also for this workshop on fundamental application of genome editing, GMO, and its regulation. I would like to also to extend my sincere appreciation to to the National Biotechnology Network of Expertise that in collaboration with ICARDA, the International Center for Agricultural Research in the Dry Areas, and AGERI, the Agricultural Genetic Engineering Research Institute, organize this meeting and bring together such a diverse and distinguished group of participants. Your presence here reflects the growing importance of biotechnology in shaping the future of agriculture in Egypt and in the region. Egypt has long been a regional leader in agricultural innovation and biotechnology, and today is an important milestone in on the road to establish genome editing technologies such as CRISPR, and because it's opened unprecedented opportunities to improve resilience to climate stress and ensure food and nutritional security for the agricultural sector. However, it's important that scientific innovation alone is not sufficient. From the discussion in the 90s and 2000 on GMOs, we have learned that we need to make sure that we have clear and science-based regulatory frameworks, such as a biosafety law. We have effective communication with the public and a strong collaboration among researchers, policymakers, farmers, and the media. Therefore, this workshop is both timely and strategic. It aims not only to strengthen technical capacity, but also to build a shared understanding of the benefits, risks, and responsibilities associated with the genome editing technology. Over the next few days, we will engage in cutting-edge scientific discussions, hands-on training in genome editing tools, and important dialogue on biosafety, ethics, and policy frameworks. I therefore encourage all participants to actively contribute, share experience, and build lasting partnerships. The strength of these initiatives lies in the network that we create and the knowledge that we exchange. For an opportunity to position Egypt and the broader region as a leader in responsible science-driven agricultural innovation. I wish us and you all a productive and inspiring workshop. Thank you.
I would like to welcome Rahmat Gabri, who is the Vice President of ASART, Academy of Scientific Research and Technology.
عليكم صباح الخير على حضراتكم. شكرا جزيلا للاستماع. Actually, I have my speech in Arabic, but I will say it in English. No problem. No, it's no problem. First of all, I want to thank Professor Naglaa for her great efforts in the National Network for Biotechnology Experts. Actually, this is not the first hands-on training. I think it's the third one. Okay, and thanks to ICARDA for the continuous collaboration for the all training. Actually, we are talking about the climate change. We are talking about the most problem, many problems face the agriculture sector. Biotechnology is one of the most important technology to facing the climate change, facing the water scarcity, facing the food security. One of the most important tools in biotechnology is gene editing. We are talking about today. We have a lot of debate about the gene editing. It's GMO or non-GMO. Some countries now approve. I think recently the EU approved that gene editing is not a GMO. It's GMO is a marketing. GMO is a facing a lot of problems. Gene editing will approve as a non-GMO or will be certified as a GMO. I think we need a lot of discussion, especially we have expertise, we have a scientist, we have a political in this today, in the first day or in this discussion. I hope to have a real vision to certify the gene editing as a GMO or non-GMO. Thank you all. Wishing you a successful discussion and success hand-on training. Thank you.
بس احب اذكر ان دكتور احمد اساسا ساينتست وبايوتكنولوجيست وبيشتغل على النخيل، فهو يعني جاي في المكان الصحيح. دلوقتي بنرحب بالدكتور مجاهد عمار مدير معهد حصيل الحقليه، اتكلم in behalf of دكتور عادل عبد العظيم. اتفضل.
بسم الله الرحمن الرحيم. Good morning to you all. It's a pleasure for me to be here on behalf of Dr. Adel Abdelazim, the president of ARC. He's conveying his wishes in regards to this workshop. Thanks, Professor Nagla, for the invitation. Welcome, Dr. Baum, Dr. Ahmed Lina. It's a pleasure being here today. Actually, I will talk as a plant breeder because if we are talking about genome editing, then we are trying to solve the problems of plant breeder. And imagine if a plant breeder stayed 10 to 15 years developing a variety, and due to the climate change scenarios that we are witnessing today, the resistance has broken down or it becomes susceptible by any other reason. So he lost the 15 years of continuous efforts. So if there is a means to restore this resistance or this trait, this is a dream for every plant breeder working on this field. Egypt is having a remarkable progress in crops. Particularly, we are ranking first in rice per production per area, with 10 tons per hectare. Maybe the first also in spring wheat, with over than 7.15 tons per hectare as a national average. And maize, we are fifth globally. So we are ranking in a very good position. But what hurts the breeders is that their varieties, after being released, there's some coincidence of breakdown of the resistance or whatever the stresses is there. It makes a big problem for this variety. And we have some examples in wheat, for example, Saka 69 was one of the, or 61 also, was a very good variety. But unfortunately, the disease resistance has been broken down and we lost this variety. In rice also, Saka 101, Saka 104 also break down by blast. So these important traits, when it is breaking down, the variety actually loses its value in the market. And what we need is to restore this specific trait. We don't want to change the type. Here, maybe Saka 101 rice variety is a very good example because it's ideal plant type, the best variety that we have developed in Egypt. When it is broken by blast fungus, the farmers are still growing it till today because of the quality, because of the yield potential, because of so many things. So if we have a mean to restore this resistance, this will be great. And in the same time, keeping the merits of the variety working as well. So I'm very optimistic that after a long years of debating on GMO or non-GMO, and whether it is the right direction to go or not, now we have come to intermediate technology that could really be the solution. Genome editing, it is, I consider it is innovation of site-directed mutagenesis. You are just knocking on the particular point, changing it, and look for the phenotype that you are looking for. I will not talk too much. I wish these discussions to be fruitful, and I will also copy what my colleague Ahmad said, that the presence of the scientists, makers, and companies also who are working very extensively on this direction would be very helpful actually to come up with a platform of whether to go in this direction strongly or we have to wait, or what would be the scenario in the coming years. The National Biosafety Committee really needs to actually address this issue very well, because we are in a very challenging environment. Egypt is a totally dry country. Water scarcity is imminent. So we are looking for a technology to solve these problems. And I think gene editing could be the possible solution. Thank you so much. And I wish you a very successful event. Thank you.
Now I would like to welcome Dr. Lina from AHEI. First of notice, I just informed her that she's going to speak. Thank you very much.
Okay, good morning, everyone. And first of all, I would like to thank Suasun and Regla for inviting me here. This is my second time to Egypt, and I'm already in love with Egypt. It's so beautiful country where I was always dreaming to come. And so this is, I will also say, I will just echo everybody said, like, gene editing is like a new tool, but it has a lot of power. But what I will say here is this is not silver bullet. It will not solve it of its own all the problems. It's a tool which can be complemented with other technology which everybody is already using. Like, for example, breeding, right? So breeders are already using the breeding. This tool can come and accelerate the breeding so that, if you cross and you go, you don't need to do several. So that's what this tool is. And many countries are already benefiting with this tool. Then why Egypt should lag behind? Because Egypt is also has, as I said, a lot of potential, not only to self-sufficient, but to feed others, because you have a lot of land. If the drought problem can be solved, then the wheat and the maize can feed others, who are right now struggling for those type of crops. So in several countries now, I will show in my presentation that what the regulatory framework. There's already several countries where gene-edited crops are treated as conventional if they have no foreign gene integration in them. But it's also happening on the African continent. There are 5 countries where the regulations are in place. Not only the regulations, I was actually involved when the regulatory guidelines were, the Kenyan regulators were developing the guidelines in Kenya. So I will be more than happy if I can be of any help when Egypt is developing those guidelines as well, because I do have experience from those. But what I will say is, like, in Kenya, when in 2015, my lab started working on gene editing, and when I went to talk to the regulators there, they said, like, we don't understand. Can you come and give a presentation? What's the technology is? What? So, I went, I even gave the presentation to the board members to understand. So now the guideline was there. And I'm so happy to say that, last week, we got approval of the first in-house developed gene-edited product, which is now categorized as non-GM. So it's like a similar to the conventional. So this is gene-edited banana resistant to the bacterial disease, and which is now going to the field as like a conventional variety for the testing. So no confinement, no biosafety regulation as GM. So that's the benefit, like, okay, it can be accelerated using this technology. But in comparison to I'm not saying GM is bad, because GM also has a lot of benefits, but there is a lot debate going on. But also there is a heavy regulatory cost there, which can be reduced if you can, your gene edited components or the crops can be treated as conventional. So developing a regulatory guideline for, like, very clear is very, very important. And I hope, like, Egypt will have that guideline soon, so that the projects we are working together on date palm. So when the date palm, gene edited date palm varieties are developed, we are thinking like those can be tested, but also the growers here can benefit with those varieties. So with that, I will stop and I'll wish everyone the successful workshop. Thank you.
Thank you, everybody. I just want to mention that today is divided in several themes. First, we're going to have an opening with the people who funded this event from ICARDA, from the people from the project, from Repal Vivo, and from people from ICARDA. Then we'll give a part of our core scientific background, very simple. We don't want to go in details. Then we have the regulatory part, and finally we have the communication, because communication is very important, how to can deliver the message to the people and for the stakeholders. And at the end, we have an open discussion. We hope that everybody from you can join the discussion so we can get with a recommendation, a food recommendation about the editing, how we can implement it and take the message to the decision maker. Dr. Sasan would like to give an
أوكي. بحب أشكر المنصة الكريمة على الكلمات الرائعة واللي وصلت تقريبا مضمون اليوم اللي حنعمله النهارده. دلوقتي أنا أدعو جميعكم للجروب فوتو، بعدين عندنا الكوفي بريك، وأفتر وبعد هيدا رح نبدأ بالسيشن البرزنتيشن from inside and outside online from the people. Please, you are all invited for the group photo, then for the coffee break.
There is a small correction. We'll take photo first, and then Michael will give his presentation before coffee break. So about that, we have to postpone the coffee break after Michael's presentation. Okay, thank you. I will
That's okay.
All the that's what we all do because we make a living.
خلاص من عنده. كده. كده خلاص عارف.
Dr. Mugib,
Yep.
you turn Dr. Mugib to present your file, please.
After. Okay.
Now, Dr. Mugib, if you kindly.
Hello, Dr. Mujib. Good morning. Now, Dr. Muhammad will introduce you, and then you will start your presentation. You will be the first. Sorry
Okay.
for the changing we made, but yeah, forgive us.
No worries.
Dr. Muhammad will introduce the speakers or presenters.
السلام عليكم.
Dr. Mujib, can wait for a couple of minutes, please?
Sorry, can you hear me?
Can we wait for a couple of minutes, people?
Okay, sounds good. Sounds good.
Dr. Mujib?
Yes, I can hear.
Okay, we are going to start.
Okay.
Okay. So my name is Mohammed Saad. I'm a senior researcher at the Agricultural Genetic Engineering Research Institute. And I have a great pleasure introducing Dr. Mujib Stanekzai, a CARDAS project manager for research for development and the interim UAE country coordinator. With over 23 years of experience across the Middle East, North Africa, and Asia, Dr. Stanekzai is a seasoned leader of large-scale programs, having directed portfolios exceeding $270 million, securing over $52 million in new funding, while leading multicultural teams of up to 600 members across complex environments. In addition to his project management expertise, Dr. Stanekzai brings a unique perspective to his work, holding a medical degree from the Kabul Medical Institute, in addition to being an PMP certified professional who had a master's degree in comparative local development. He's an expert in aligning scientific research with national agricultural priorities. He currently manages the consortium for the Consortium for Red Palm Weevil Control, of which he will give us a quick overview. Dr. Stanekzai, the floor is yours.
Thank you so much, Mohammed. It's a pleasure to be with you. So, distinguished guests, respected colleagues, ladies and gentlemen, assalamu alaikum wa rahmatullahi wa barakatuh, and good morning to everyone. I don't know if everyone can hear me clearly. Yeah, it is a great pleasure, like I say, to welcome you all to this session. I'm honored to have the opportunity to share with you an overview of the Consortium for Red Palm Weevil Program Initiative and our collective efforts to address the challenge of red palm weevil through decision-based and collaborative approaches. The Consortium for Red Palm Weevil Control is a flagship research for development initiative led by CARDA under the CGIAR systems. It brings together governments, institutional research centers, national agriculture system, the private sector, and the growers to address one of the most damaging transboundary pests affecting palm system worldwide. When it comes to the global challenge, it is a global challenge. Red palm weevil no longer is a localized problem. Its rapid spread across continent has turned it into a global biosecurity challenge, affecting production system, trade flows, and national quarantine regime. Once infestation symptoms appear, palm are often already lost. This makes prevention and early detection not just preferable, but essential to the overall approach. When it comes to why Consortium for Red Palm Weevil Control, part of this you will hear later on from my colleagues, Michael Boom and Dr. Lena and Dr. Sousan, which is contributing to one of the components of this program. But overall, the consortium was created because red palm weevil cannot be solved through isolated intervention or short-term responses. The pest crosses borders and institutional mandates. Effective control requires an integrated system-based approach that brings together prevention, intelligence, field practices, and governance all together. When it comes to a pioneering global consortium model, what makes Consortium for Red Palm Weevil different? Consortium for Red Palm Weevil represents a new model of collaboration. It is not a simple project or a technology pipeline, but a global platform that aligns research excellence with national priorities and field realities. This structure is essential for addressing a pest that threatens the global food system and trade. So it integrates CGIAR centers, National Agriculture Research System, advanced research institutes, private enterprise and technology developers, farmer, municipalities, and growers all together as a one team. When it comes to vision and strategic approach, the consortium is organized around a simple but transformative logic, and it is early detection, early warning, and early action. This is supported by science-based intervention and digital tools that allows stakeholders to act before palm are lost. So there are key pillars that I will talk about also in another slide. And that key pillar is digital innovation and data-driven decisions, bio-based and biotechnological solutions. On the biotechnological solutions, you will be hearing about it all day today. Good agriculture practices, strong policy, and phytosanitary system is another pillar that this slide will probably explain it a little bit more. So the program architecture consists of 5 interlinked work streams. The 5 work streams allow the consortium to leverage specialized expertise while maintaining system-level coherence. The technical steering committee under the governance structure ensures scientific rigor, alignment across partners, and critical decision-making. And also under governance structure, the funding committee that we have for the program is ensuring that the program continuously receives funding and gets implemented. So the 5 work streams, again, are climate resilient bio-based red palm weevil control, biotechnological innovation, RNAi, genome editing that you will be hearing about this in this workshops, microbiome, and also the 3rd pillar is digital innovation and big data analysis. The 4th pillar is good agriculture practices. And then the 5th one is integration, adaptation, and scaling pathways. When it comes to integrated, or we are calling it solution bundle, so a key innovation of the consortium of red palm weevil is the concept of solution bundle. It is real because real impact comes not from individual technologies, but from integrated package that are scientifically validated, operationally feasible, and socially adoptable. And this bundle combines technology, digital intelligence, management practices, and institutional and policy support all together. So when it comes to what workstream delivers, workstream 1 to workstream 5 generate innovations and knowledge. Workstream 5 integrate and validate these contributions into a deployable packages, ensuring that solutions actually work on farms and within national system. So each one of them is contributing a very niche technical and scientific area. For example, workstream 1 traps chemochemical by pesticides in biological control. Workstream 2, RNAi, microbiome-based control, long-term resistant pathways. Workstream 3, sensors, satellite, and drone imagery, all AI-driven analytics and dashboards. And workstream 4, good agriculture practices packages, sanitation, irrigation, nursery certification. And workstream 5, the bundling, the field validation, socioeconomic viability, and also scaling the pathways to countries. And when it comes to the overall program and the program, the current landscape of the program, UEE is used as the flagship innovation hub from field validation to global impact. But we are now expanding that because the UEE in Egypt, they are both play a strategic leadership role, providing an enabling environment for innovation, testing and validation. Listen, learn here are critical for adapting solutions to other countries facing similar red palm weevil risks. So in this map, you will see the sites that are selected in UAE for our intervention, and also the sites that are selected in Egypt from with the support from both ARC and DRC that we will be working on and testing these bundles. Overall, when it comes to phase 1 progress from 2025 until now, and also going to phase 2, phase 1 focused deliberately on readiness rather than robust deployment or rush deployment. This ensures scientific rigor, system alignment, and credibility, laying the groundwork for durable impact in the following phases that we have in front of us. So overall, there are key areas in the slides that we have focused and we are working on, and then going into our next phase. And then when it comes to early value and strategic impact, even at this early stage, Consortium for Red Palm Weevil Control demonstrates strong value for money. prevention-focused systems, protect high-value palm assets, and reduce long-term reliance on pesticides that we have in front of us. And then when it comes to overall looking ahead, so the integrated approach that I have talked about before, and also spanning science, digital innovation, and system-level adaptation is exactly what our upcoming panel will explore. Our integrated red palm weevil management can move from pilot to policies and lasting impacts. The panel that we'll be talking about more is about the second pillar of the program, but the slides that I have earlier is about overall red palm weevil program that we have. So from foundation to the field transformation, it has integrated bundles deployable and field tested. We are working on early detection, early warning, and early action digital platform and it's fully now operational. We also have scaling through national system and policies, and we are positioning for red palm weevil control as a global biosecurity priority. So this is overall in a nutshell about the red palm weevil programs. I'm sure that you will be hearing a lot about the overall work stream 2 level activities that we have in front of us, especially on genome editing. And again, thank you all so much, and thank you for the opportunity to give you this short overview on the red palm weevil program. Thank you.
Thank you, Dr. Mujib, for the nice presentation. On to the next speaker. It's a privilege to introduce Dr. Nagla Abdallah, Professor of Genetics at Cairo University, and a prominent figure in the biotechnology landscape in Egypt, named as one of the most influential people, 100 most influential people in biotechnology. Dr. Abdallah leads the National Biotechnology Network of Expertise and serves as the editor-in-chief of the journal GM Crops and Food. With over 80 high-impact publications, Dr. Abdallah has conducted advanced research at prestigious institutions, such as the Scripps Research Institute and the Donald Danforth Plant Science Center. Her work has been instrumental in shaping the scientific, regulatory, and ethical work frameworks of genetic modification, genome editing, and biosafety in Egypt. She also had a talent for bringing institutions together, having served as the chief of party for a USAID initiative that united 4 American and 5 Egyptian universities into a center of excellence for agriculture. Dr. Abdullah is the scientific advisor for the biotechnology workstream activities within the consortium of Consortium for Red Palm Weevil Control here in Egypt. She will give us an introduction on the basics of gene editing workflow. Dr. Nagi.
Everybody, so it's me again. So I'm going to give you like a brief about the fundamental issue about gene editing because we have an expert going to talk in details about that, and we have a training workshop coming in the next 4 days. So this is the main topic I'm going to cover during my presentation. I'm going to give an overview about agriculture in Egypt. I'm not very qualified to give that. We will have an expert people from the ARC. They can give better than me, but I'll try to cover it. Also overview about gene editing, application of gene editing, regulatory issue, concern about gene editing, and conclusion. So agriculture in Egypt, as you know, that Egypt is mainly an agriculture country. Agriculture in Egypt is a vital economic pillar, contribute about 14.5% of gross domestic products. Correct me if I'm giving a wrong account. It's employee 28% of the workforce with production in citrus, potato, cotton, and different crops. It's central along the agriculture is central along the Nile. Heavily relied on irrigation. Agriculture exports 6.24 million pounds by mid-2025. The total cultivating area is about 9.72 million per day in 2023. So agriculture they face different challenges in Egypt, mainly because of the population, the growth of population, and increase in food demands and climate change, water, and land scarcity. So as we know, agriculture in Egypt is pioneer. We already started using biotechnology several years ago, 2 decades before, and it was one of the first countries that used gene edited genetic modified in the region, also in Africa. But unfortunately, it was banned, and here we can see that we already have developed BC cotton, but unfortunately this product was not implemented. So I want to say that Egyptian scientists is pioneer in the field of biotechnology, but they need a chance to implement their product to get benefit from this product. So we need to, for sustainability, we need to close the gap for yields and especially in staple food. We know that we have a big gap in the field of in the food to offer the people, and also we need to use new technology to cover this gap. So breeding, it's very important, but it cannot solve this gap by itself. It needs another innovative technology such as gene editing to help to close this gap. So we're going to give like an overview about gene editing. As we know here, we can see that GM already started very early in 1994. The first product was 1990. The first product was in 1996 for cotton, corn, and canola. Different country, of course, United States was pioneer in this field. But gene editing, as we see here, it started late. We have commercialized the product in 2016, the already product in the markets. Until now, we expected by 2035, this product will be covered in all over the world because it's important and how it can solve the main problem and increase our income. So as we see here, these different crops that have been developed by gene editing. Most of them are research, but not all of them in the market because a lot of difficulties. We're going to talk about it. So as we see here, gene editing is highly speed developed. So we have a lot of publication, more than 1,000 publications in gene editing in crops only. This is the European website for gene editing database. So gene editing is, as we said, it's not new. It's came out from mutation. So the idea of gene editing that we can use mutation directed in a specific site to reach our goal. As we know, mutation, we can use radioactive mutagenesis and so on. And this can be random, can cause a lot of problem. But we can induce mutation very precisely using gene editing. So gene editing, as we said, is not new. In the beginning of 20th century, mutation were accelerated using chemical and radiation. Right now, we use precise mutation, a site direct mutation in the target site that we want to improve to reach our goal. So gene editing, we can use different technology like CRISPR, TALENT, zinc, and so on. These are all of the technology that are the target of them is to induce a cut in the DNA and allow the cell to make the repair. So, but of course, CRISPR are the main, the top most popular one used for gene editing. So as we said here, that once the mutation happens in the DNA, the cells cannot leave this mutation by itself. It has to be repaired. So it's either direct repair by rejoining them, but it induces mutation, or we can use it to replace the target part of the piece. So we can get benefit from the cut of the DNA and introduce the part we want or make a mutation in the target site. Gene edited faces a lot of generation. The first generation is using the CRISPR-Cas9 and the Cas12. This is for direct mutation, cutting the DNA double-stranded and so on. The second generation, it was developed from the first generation. They can use it for base editing. That means that we can change one basis by another base that we are targeting them. And this is very precise process. And also for prime editing. Prime editing also is very precise, as we can replace or enter very short sequence in the target site. So the second generation is very precise modification in the genome. We locate the part we want to change and we change it accordingly using these techniques. The third generation, we can use epigenome or transposon on the recombinase. This epigenome meaning that we don't change the DNA at all. So the DNA is stable, but we can change the activity of the gene. So we can make the gene more active or repress the activity. This can be used by modifying the structure of the DNA or the histone. So if we do the sequencing for edited epigenome edited plant, we cannot find any modification. But only we can see that the gene expressed is modified. Also transposon, we can use it for entering a very large part or deleting a very large part in the genome. So all of this in the third generation. And during the course we're going to cover, we will take more details about the different 3 generation techniques. So as we know, genetic modified organism or GMO face a lot of problem because we enter a new gene and we can enter the gene randomly and so on. So scientists came out with gene editing so they can modify or can let the people more accept the edited plant, and it will be easier to commercialize it and get through the regulation. So there is different methods for getting non-GM crops. We can make the modification without having a foreign DNA in the structure of the genome. We can use a ribonucleoprotein because the CRISPR need only the RNA and the protein. So once we introduce in the cells, they do the editing and then they will disappear. So we can use it with protoplast. We can use transient expression with Agrobacterium, mutated Agrobacterium or Agrobacterium aeruginosus. This will not enter the DNA into the genome. We can use also segregation. So if we use transgenic, this next generation will get rid of the transgenic plant. Or we can use viral vector. So the viral can carry the RNA and the protein and make the editing. And then after that, we get rid of the virus in the next generation. So all this technique can produce a plant without any foreign DNA. And a lot of countries allow edited plant without DNA to be named as non-GM and can be commercialized directly. So why do we need to develop a genome editing protocol? Because it will be accessible for the farmers and for the public to be cheaper than GM, more acceptable by the consumers and the regulators, and can be used for mitigating climate change and support sustainability of agriculture in different countries. Of course, it will can address some sustainable development goal for the the different countries. So genome editing for crop improvement can be is a group of technology. It's highly developed technology, can be used for precise modifying the expression of the gene, not necessary to modify the DNA, but modifying the expression. So we can, if you want to switch off the gene, we can switch on. We can change the gene to be like the wild type plant. We call it cisgenetic. Cisgenetic meaning that we can do it like the breeding. Breeding, we can do crosses to let the gene like the wild type plant. We can use gene editing to make the modification for the gene presented in the wild type plant without doing the breeding. So it's very precise and very useful and easier to do compared to GMO. So it's become very important for molecular revolution and for modern breeding. It's also it can be used for any kind of organism, starting from the bacteria to the human. Unfortunately, some people use gene editing for the human, for developing people, like in China, but it's forbidden. They put a restricted rule, so they prevent the developing babies with edited genome. And of course, as we said, epigenetic or epigenome editing, it can be developed without changing the DNA. So genome editing has a potential value because we can, as we said, I think this is a repeated one because we said about the Sustainable Development Goal and mitigation. The application of gene editing can be applied, as we said, starting from agriculture, for environmental management, for biomedical research and gene therapy, for livestock, and for industrial outcome and challenges. But we're going to mainly concentrate on the agriculture and food production. For agriculture, we can say that they can be used for developing resistance against various biotic and biotic stress. So we can see that a lot of products came out with tolerant for salt and drought and heat and different abiotic stress, as well as biotic stress like bacteria, virus, fungi, and nematode, and so on. So all of that can be developed by using gene editing. Also, we can use it for enhancing yield and nutrition in the different plants. Here are some products that are already commercialized and approved for foods in different countries, mainly in the United States, in Canada, and Japan, India, China, and Philippines. So these are the products until now, what I can make a screening for that they already in the market, and they name it non-GM. It's already in the market. If you go to Japan, you can find tomato written non-GM edited plant. So this country allow to commercialize this product as long as they don't have foreign DNA in this product. They don't have to go through biosafety regulation. So as you see here, a lot of product in the United States, this is the biggest country that develop and commercialize edited crops, even in animal and in fish and so on. Also in Canada. Canada is the second, followed by Japan, India, and China, and Philippines. For Africa, we have a lot of projects going on. So this is a survey done by a project with NIMPAD. It's just coming out for publication. As we see here, the biggest country who developed edited plant is Kenya. Of course, IITA is the main source of edited plant. They have a lot of work there. I was honored to visit them and see a lot of very new work going in their lab. So Kenya is the biggest one. They have work not only in the plant, but they also have in microorganism in livestock and so on. Followed by Egypt. Egypt, as far as I can make a survey, we have 8 projects for edited plants, maybe more, but this is what I can get from my connection, from the publication and from the direct connection that we have 8 edited plants in Egypt, mainly in maize, in corn, in chickpea, and so on, as we're going to see in the next slide, followed by Nigeria. Kenya and Nigeria have a law going on for edited plants, so they allow commercializing or approving the edited plant without going in regulation, followed by Ethiopia and so on. This is all the country that we make survey for them. The survey was done only for 16 countries that mainly they use gene editing. So other countries like Tunisia, Morocco, Tunisia, Cameroon, and Malawi, Senegal, they don't have any project. Morocco has just started working on edited plants, and they have running on work for students for register for edited plant, and they are also writing draft for genome editing regulation, and I think they will approve before us because I take it very seriously. So for other the different kind of gene editing, as we said, for crops we don't have in Egypt. We only have it in Egypt. For livestock we don't have it. For microbe and for agroforestry, this is the percent of the product in different countries. For Egypt, as we see here, we have, as we said, we have 8 projects mainly funded by USAID, like some of them are already completed, like in wheat. We have one done with Riverside, with the United States, joining the Cairo University and the United States. Also, there is one with the Kushrin from Agiri and from Florida University. Those are already completed. There is another running project like in chickpea. This is with the collaboration with Cairo University, Agiri, and Picarda for drought tolerance. We're using 2 different genes, very important to get rid of the or to get plant tolerant to drought resistance. And the work is going on, and we hope to get the result very soon. Other one is for maize, and this one also for drought resistance. We use not only gene editing, we use also for activation. So we use 2 different advanced methods to get plant tolerant for drought in maize. And also there is work is going on in the publication from potato and from Pitonia and the rice. All of them are participated by Egyptian scientists, and they have their publication there. The end. So as we said, in animal, the amount of product is very low, but there is some of them already in the market. So as you see here, these are in the market, the one in here. The other is going on, still they have work, but not approved yet. So as you said, it's not only for crop, but also for animal. So I'm not going to go through regulation very so much because we have 2 experts going to talk about regulation, about gene editing. But we have a lot of people have concern about gene editing for crop improvement because we have a bad communication. That's why we organize different communication workshop with private sectors, with Alliance of Science, and with the Academy of Science, to just deliver the message and explain to the people that gene editing is secure, and a lot of countries is implementing them. So in the previous, the gene editing was divided in the side-direct nuclease 1, 2, or 3, but right now the European, they have different vision about classification. So for SDN1 and 2, mainly they don't have foreign DNA or have very small foreign DNA. They can produce by mutation, by base editing, and so on. SDN3, this one, they have a bigger part of the introduced DNA or introduce the whole gene. So most of the countries that already have the law, they considered SDN1 that is non-GM, SDN2, they take it case by case, but as long as it does not have a big DNA, they can approve it. SDN3, they treat it as GM. Of course, it has less regulation, but still considered as GM, and they have to go to the committee for GM. For the conventional, of course, it can be produced directly. They don't need any kind of regulation. So for the, as we said, the European, they classified into a new genomic technology. They name it in NGT1 and NGT2. NGN1, that meaning that does not have any foreign DNA, and they consider also the cisgenic as non-GM. Cisgenic meaning that we can develop the gene that, like the wild type, it can be produced by breeding. So they consider this as non-GM, except the one that develop the pesticide or resistant to the pesticide. So although it's safe, but they said if we develop plant resistant to pesticide, that means that we will introduce pesticide in the media for a big amount. This will cause mutation. So they have a vision that if we use edited plant does not have foreign DNA, but they have resistance to pesticide or develop pesticide product, they consider it's not allowed to be considered as NGT1. The NGT2, of course, it has a foreign DNA and is considered as GM products. So all of that will be covered by experts for formulation. And can we see here, we have North Africa, Latin America. This is an example of product they already approved in Asia and in Africa and so on. This is the different country that already have the law. And here we see a lot of presentation about the regulation in different countries. This is one is 2026, showing that the green one, they approved commercializing gene edited plant without foreign DNA, without the regulation, and they already have regulation to follow up. The gray one, they don't have law. So Egypt, as we said, we don't have law, but unfortunately, as you see the spot here is Israel, they have the law and they approve the GM, but other countries, they don't have the law. So we face a lot of challenge and concern, most of them for technical and safety challenge, like off-target effect and the delivery method, mosaic, because some of the plant, part of them are edited and other not edited, long-term effect and so on. For public perspective, as we said, we need to deliver the message for the public because they don't know about gene editing. Most of the people, the consumer and so on, they have the wrong idea about this. So I think Sheila will cover this part for communication, and she will explain how we can overcome this problem. For environmental and by safety concern, a lot of people from the environment have concern about gene editing. For the regulatory challenges, as we said, this is the big challenges in our country. We have good scientists, we have a good background, we already make a lot of training course, but the problem is the product we produce, where are we going to go? Is it going to be in the shelf, or we are going to implement these products for the public? So of course, ethical concern regarding gene editing in human, this is the main problem. It's not allowed to use it, all the country to use it in human. As conclusion, this is the last slide, sorry for taking too much time. Gene editing is considered as an efficient tool for improving biotic and abiotic stress tolerance and food security. Clear, ethically grounded guidelines with scientific based regulation are essential to accelerate the commercialization of gene editing products. Gene editing techniques will have a revolutionary breakthrough to better benefit for our lives and the world. Thank you very much. I think we can keep the question for the end. خلي أسئلة في الآخر. الأسئلة لو حد عاوز يسأل ولا يسأله دلوقتي. Anyone want to ask? ممكن في الآخر؟ ماشي أحسن.
Thanks, Dr. Nagla. Then prepare this to Dr. Lina. So it's a distinct honor to introduce Dr. Lina Tripathi, the director of the Eastern Africa Hub and the Genetic Innovation and Biotechnology Program lead at the International Institute of Tropical Agriculture in Nairobi. With over 26 years of expertise, Dr. Tripathi is a global leader in the genetic improvement of staple crops like banana, cassava, and yam, a fellow of both the American Association of Advancement of Science and the African Academy of Science. Her work bridges the gap between high-level molecular biology and practical field solutions for sub-Saharan Africa. She's a strong proponent of the science to practice approach, a philosophy that ensures her high-level research does not just stay in the lab, but translates into a real-world solution for farmers globally. Beyond her research, she serves as an adjunct professor at the University of Queensland and sits on the advisory boards of the Cornell Alliance for Science and the Norwich Institute for Sustainable Development. Dr. Tripathi is the lead scientist of the biotechnology work stream within the consortium for red palm weevil control. She will show us how gene editing can impact agriculture in Africa.
Thanks for the kind introduction. So I will actually talk about the application of gene editing and how it can benefit in terms of the agriculture in Africa. So, I don't need to say much about it, but you know that currently the major challenge globally in agriculture is how to feed the rapidly growing population, which we need actually about 60 to 70% more production if we want to feed in order to feed everyone. But on top of that, climate change is actually affecting or creating the greatest challenge, because now it has like a lot of negative impact we already started seeing. And it's not only the crops, because the pests and pathogens are also becoming more aggressive. So, we have to also think in that direction. So in terms of the agriculture in Africa, as you know, more than 60% of the population in sub-Saharan Africa are the smallholder farmers. And then about 23% of the GDP comes from agriculture. So agriculture is quite important, yet the Africa's full agriculture potential remains untapped. Africa has potential to produce 2 to 3 times more because of the availability of the land and everything. And then that can add 20% more to the current output worldwide. So that means they have potential not to feed themselves, but to also feed others in the world. But we need the sustainable agriculture, and that's where the problem is. That currently the yield gap is quite huge, and when I say yield gap, means what the potential is, and then what currently the farmers are getting. The gap between that is quite huge, and then in order to go like 60% more production, we have to close that yield gap, and that yield gap even get more because even after the harvest, the post-harvest losses are also there. So these are the things which we have to think for the future if we are talking about sustainable agriculture. So why gene editing then? So with increasing demand for the food, but the resources are limited. It's not everywhere there is like increase in land or the water. And as I was mentioning, that we need to close the yield gap in a stable crop and enhance food production. So that means, we have to use all the tools available in our toolbox, and then including the new breeding innovation tools, which is genome editing, but we also need to include like high throughput phenotyping, digitalization, farm mechanization, and as well as the climate smart agriculture. And gene editing is actually one of the very powerful tool in our toolbox. Is the similar thing like, you know, if the car breaks down, we have to use, you know, necessary tool which is required for that. So that's the same thing in the agriculture as well. And then, you know, new tools are keep on adding to the toolbox, and we have to see how the best we can utilize them. So as Dr. Nagla already mentioned that, you know, gene editing is basically not a new tool. It is basically a technique which can be used very precisely and efficiently for making the changes. Basically, if you want to see the gene editing is like the same way, like, you know, you edit your Word file, right? You know, you go delete or add or replace. It's the same thing you can do in the gene editing in the genome of the human. Nature has been doing this from the long time. So, you know, nature used to edit and then farmers go and select, but, you know, that's a very slow process. So that's why in the 20th century, people started using mutagenesis. They used radiation and as well as the chemical mutagenesis. But those mutagenesis are random. So you don't know where exactly is happening, but, you know, you also see unintended effect of that because it can happen random. And then later on, the technologies have developed for more precise, like talents. These are the technologies, but those technologies were still quite complicated because the editing through the talents and the meganucleases, the protein chemistry is very difficult to use it. So then later on, when CRISPR came, which is, the name is very complicated, very big, is clustered regularly interspaced short palindromic repeats, but in reality, the technology is very simple. And so that's why, you know, it's easy to adapt, and it has become actually the most popular genome editing approach right now. Everybody is started using CRISPR because it's very easy to adapt. But then, you know, it doesn't stop there. You know, if you say CRISPR, like, you know, initially it was only knockout, then now is a base editing, prime editing. So, you know, every other day you see like a new technology is coming up. So, you know, we have to be always remaining on our toes, you know, to learn the new technology. So there is a lot more new breeding tools are happening. So gene editing has a lot of potential. I will not repeat what Dr. Nagla mentioned about the like what can be done in terms of the agronomic. But what I'll say that this technology is so powerful, and that's why these two wonderful ladies got the Nobel Prize in 2020 in chemistry, Jennifer Doudna and Emmanuel Carpentier. So currently, actually, if you see these bars, you see very, very slight orange on one of the bar, very thin line actually. That's the only thing which is either commercialized or ready to commercialize. But then all the green part, like you see, this is all in the pipeline. So is for using this technology in the crops, majority of things are actually in the pipeline. But what I would also like to say here, the upper bar is for the private sector, and then the bottom bar is for the public institutions or some new startups. So you can see it's quite a lot is going on in the public, in the academia, in the universities, in the public research institution, and the startup. What does it show? It shows that, you know, is much easier and not that expensive that, you know, only private sector can do. So that's what I wanted to emphasize with this. These editing technologies are already applied in more than 40 crops across 25 countries and addressing the agronomic, food and feed quality. And so this pie chart, you can see there are a lot of traits which you can tackle in using these technologies. These are the only very few products which are either in the market or approved for the commercialization. All the big list which Dr. Nagla was showing, these are like in the pipeline or approved at the first level for the field trial, but not in the market. So this high oleic soybean oil is actually already in market. Then in Japan, released these tomatoes. Then there is a conscious green, the salad, which is developed by a small startup companies. And here I would like to say that, you know, the rice you are seeing, this is developed by a public institution in India. So 2 rice varieties developed by research institution, actually they are now approved for commercialization in India. One of them is actually for drought. So this is very latest. this is the publication coming out is one of our paper in Nature Plant is in press. I think it should be out next week. So this is the current, very latest, the landscape for the regulatory for the considering the genome editing. And here the green are the countries, the shaded in green are where the regulatory guidelines are already in place and there are no restrictions. So it's case by case and is more on the product based. The yellow are the ones where there is already discussion going on. EU has approved, as almost all of you know, but it hasn't gone to green yet, but it will go, I think, in July when everything comes up, it will become green as well. So South Africa, in Africa, actually, South Africa is the only country where they treat the GM and the gene editing the same. You can commercialize the gene edited product there, but it will be similar to the GMO. So, you know, it's a heavy cost related to the regulatory approvals there. But in Africa, there are already 5 countries where the regulatory guidance is approved. So Kenya, Nigeria were the first. Now Ethiopia, Ghana, and Malawi also joined, but there are several other countries where this discussion is ongoing. So there are, if we can compare, like, you know, there is some regulation and the policy support for the gene editing in Africa, but there are few challenges as well. People who are interested, I will also suggest to read our paper in Nature Biotech, which talks about that. So in the regulatory shift, we have seen that, you know, moving away from strict GMO framework to more adopting like a product-based, a case-by-case regulation of the gene edited. So that's where we see that Africa is shifting on, and, you know, much more strong support from the regulatory agencies in Africa. So gene edited crops without any foreign DNA inserted in them are treated as like a conventional. So that's very good. In terms of the policy support, AU NIPAD is supporting in harmonization of the policy. I'm also very closely working with them because their goal is sustainable agriculture and the food system. So that's a strong support we have there. But still there are a lot of challenges in Africa. The major challenges is the limited infrastructure. So, you know, not every institution has the infrastructure to do these type of technologies. There are funding constraints because majority of the institutions depend upon the external funding, and which is actually day by day reducing. So that's a constraint. And then the low public awareness and the communication gap, because many times when you go and talk about the gene editing to the public cannot differentiate between the GMO and the gene editing. So that's why I think as a scientist, we have also great responsibility for giving a clear messages to the public for, you know, the public awareness. So this map has become old now. There are a lot of other projects going on, but here my intention is to only show that there is a lot going on in Africa in exploring the potential of gene editing. You see, starting from Egypt to the South Africa, from west to the east, you see a lot of work going on. Definitely Kenya is actually taking the lead. There are a lot of projects which are approved or in the pipeline happening. My presentation is more focusing on agriculture, but quite a lot going on in the livestock as well. So I'll give few examples. So one is gene editing for the tolerance to the maize lethal necrosis. So this is already approved for the field trial in Kenya. They have already done several rounds of the field trial. So MLN is a viral disease which reduces yield severely, like 23 to 100% in maize. And then if you talk about financially, is about $180 million losses annually, which are actually quite big. Most of the Kenyan maize varieties are susceptible. So KALRO, which is a local institution in Kenya, partnered with CIMMYT, one of the CG Center, and one of the private sector, Corteva. So these edited lines were developed in Corteva, field trialed in Kenya, and now actually they are converting it into the hybrid varieties, which will be released. And in them, there are no foreign DNA integrated into the editing editing lines. The next example, again from Kenya, is the Striga resistant sorghum. So Striga is a wheat parasite affecting a lot of cereal crop, including the sorghum. So one of the university in Kenya, Kenyatta University, actually partnership with Corteva. So again, the editing was done at Corteva and the phenotyping was done in Kenya. So Kenya regulatory authority has again approved this as non-GM product for the field trial. And so here they have like, so even in the maize and here there is a knockout. So, no insertion, it's just a small change in the gene to make it non-functional. And this is also going on in the field trial in Western Kenya. So IITA's position, because IITA is actually, we are leading the gene editing in mainly in sub-Saharan Africa, because our lab is the first, we started in 2015 working on gene editing. And the CRISPR-Cas9 is only invented in 2013. So you can see we are like the pioneers starting from the very initial stages. So currently that's the only lab in sub-Saharan Africa which has the full platform, starting from the bioinformatics, where you want to identify the targets, up to the phenotyping, everything in-house. We have all the facilities. So, so we have actually recently released the IITA's position statement, is on the public platform. And so we treat this as a safe and precise tool to improve the crops in a much faster way than traditional breeding. So we are trying to accelerate the breeding using this. And so we see that as a tool, not a silver bullet. And it complements the existing breeding approaches. So we use it very like, cleverly, like where we would like to use it. And IITA is becoming a regional hub for the development of the gene edited crops. We use this responsibly for societal and the environmental benefit. And currently we only focus on the without foreign genes integration. So Dr. Nagla mentioned about SDN1, SDN2, SDN3, but we only focus on SDN1 because that's very clear. There is no foreign gene integration in them. And then the regulators treat them as non-GM. And then we definitely respect the national sovereignty and regulatory framework. So we only work on the countries where the regulatory frameworks are there, unless the government is interested, and then we help them developing the regulatory framework. Otherwise, the research focuses mainly on those countries. So if you want to read our positional statement, is available on the website. So these are the different projects going on. I'll very quickly talk about some of them so that you can understand how you can apply. So we are working on the disease resistance in banana. We're also using optimizing the architecture of the plant, like yam. We are also improving the nutritional quality in some of these orphan crops like Bambara groundnut. We're working on cowpea for developing the aphid resistance and increasing the yield. We're also involved in Striga resistant maize, and then also in tomato, particularly for the drought. And then IITA is collaborating with ICARA on this red palm we will control. So I will talk about, like, for us, we don't just jump on working on using gene editing. We have, like, know why we need to use it, so that's the strong justification we have to do. So why we are doing bananas? So banana, particularly in sub-Saharan Africa, is a very important staple food crop. So they have this special varieties called cooking type of bananas, which you see here in the market, and these are the type of dishes that they use down there. So these are the things which are, like, the staple food for them, very, very similar to rice and the maize in other countries. But the crop is affected by a lot of diseases, like, there are fungal diseases, bacterial diseases, viral diseases, but also the pests like nematodes and weevil. The problem is not that the crop is affected by so many diseases and pests. The major problem is that when you go to the farmer's field, in the same field, they have so many diseases and pests. So that means, we are talking about, like, stacking the resistance to the diseases. And then because of this, one of the causes of, like, so much yield gap, as you see here, the orange bar is the potential, and what they're getting is, farmers are getting, is the purple. So that's the huge yield gap the farmers are getting, and one of the reason is actually the diseases and pests. So at IITA, we have very strong genetic improvement program for banana, including at the starting, like, you know, the selection from the existing germplasm. So we have gene bank. We are one of the few places where the people can do the conventional breeding of banana, and then we are using the biotechnological tools, like, you know, so currently we are focusing on gene editing for the banana streak virus, the banana bacterial wilt, and as well as the fusarium wilt. And then each tools like we are using has their own advantages and disadvantages, because if you are working on disease resistance and none of the germplasm has the resistance to that pathogen, you cannot do the conventional breeding because you need one of the parent to have the resistance. So I'll give like few examples to show how you can use this technology. This is very much interesting case, I will say. So banana streak virus, this is like a double-stranded DNA Bedna virus. These viruses, when they infect the plant, they actually send their genome to the plant, and then the genome gets integrated into the plant. So they become part of the plant genome. And then when when plants are, like, you know, in a normal condition, they will not show any even the disease symptom. But when the plants are stressed, then that's when this virus will take advantage, and then, you know, they get activated and the plants will start showing the symptoms. So this is major problem in the B genome of the banana. So banana originated from two diploids, AA and BB. So, you know, and mainly the cultivated varieties are triploid. So any variety which has a B genome, that's where this problem is. And then what you see, this, the red and the blue and the black, these are the viral genome, and the green is the plant. So you can see the viral genome goes and integrated sometimes in a tandem repeats, multiple repeats. And so this is a big problem because you never know, you know, the plant can be stressed in the field because of the change in the temperature or the drought. But even because it's a vegetatively propagated crop, you do like a tissue culture, you know, to multiply, and plant treats tissue culture as stress. So in all these conditions, these the viral genome gets activated into the infectious form, and then it will get. So we thought, like, okay, we can, with the gene editing, what we can do is we can create mutation in the viral genome without touching anything on the plant, so that, you know, the viral genome can be shredded. And in that case, you know, even though when the plant is stressed, they cannot come together to become like an infectious clone. So that's what we did. So we designed the guide RNA, which are like a important thing, like that guide RNA decides, you know, where this CRISPR-Cas9 can go and cut the genome. So we have designed 3, and then after that we stacked them and we delivered them into the plant genome. And in this case, the banana cell suspension, and we regenerated the complete plant. So all the plants, whether they are edited or non-edited, look similar when we saw. And then we have actually, in order to detect the mutation, because these are very small, like, you know, few base pair changes, we have to do the sequencing. So we have done the sequencing in order to detect the mutation. And after that, we have done actually the phenotyping. And then you can see this is our control plant where we see this is streaks. So this is non-edited control plants, but all our edited plants actually showed the no infection after like, you know, stressing the plant. So that has given us the proof of concept, and this was the first thing which we done in our lab to actually give the proof of concept with this technology. The second technology I'll show is like a banana bacterial wilt. This is a major problem in East Africa, in the Great Lake region. And then it's all the countries which this is actually the major banana producing area, and the farmers are really suffering. The losses can be up to 100% in the field, because once the disease is there, it wipes off the whole field plantation within the few weeks time. So this is the major thing is in this case is that all the cultivated varieties are actually susceptible to the disease. There is only the wild type banana which is resistant. So there is a diploid Musa balbisiana, but farmers cannot use that because it's a seeded. The fruit is seeded, but also it cannot go directly into the breeding program as well. So what we have done is we have learned, we gained the knowledge based on this the wild type banana, and we transferred that knowledge to the cultivated susceptible variety without transferring any gene. We have not transferred any gene. We only transferred the knowledge from the wild type banana to the susceptible cultivated variety. But in order to gain that knowledge, we have to do some upstream research. So we did comparative transcriptomic analysis. So in this case, we have compared the transcriptome of a susceptible variety after the infection with a Musa balbisiana, which is like a wild type variety after the infection, to see like what is happening there. And based on that study, we identified the target genes, which we can actually now precisely edit. So based on our study, we find like a big list of the susceptibility genes. So these susceptibility genes are the genes which are responsible for developing the disease symptoms in the crop. So we tested one by one, like these about 12 susceptibility genes in banana. I will not bore you with all the study of all the 12. I will only show you the 3. So the DMR6, actually DMR6 is a very well-known susceptibility gene in other crop, the downy mildew resistance. So we have actually find out the ortholog of this gene in banana. And what happens during the infection of the bacteria, this susceptibility genes gets about 60-fold increase in the susceptible variety, but it doesn't do anything in the resistant wild type. So we know that this is a very good target to edit. So we have not edited this. Is the same thing, like we transfer this editing machinery into the cell suspension of banana, and after that we regenerate the complete plant. And we have tested them by sequencing to see the targeted mutation. And after that, we have done the greenhouse screening. So where our potted plants, we challenge them with the bacteria, both the non-edited and the edited. And you can see the pot which is wilting here is the control plant, and our edited plant didn't show any symptom. The another target gene we used was the early nodelin. So these nodelin genes are mainly involved in the legumes, in the symbiosis for the nodal formation with the rhizobium. But they are also present in non-leguminous plants, like in banana. And their role is more in the nutrient transport, but also the pathogen defense, because the bacterial and the fungal colonization happens because of this. So this is a big gene family. We don't want to knock out all the early nodelin gene, because in that case that will affect some other plant. But we can precisely, with our work, we came to know that is only one nodelin 3 which is involved in banana in this the pathogenesis. So we only went and we actually knock out that gene, precisely that early nodelin 3, and then all other nodelin 3 are all other nodelin genes are still there to do the other function. And then we have done the same thing, and in the greenhouse we also saw very good resistance with them. I think this is most important, because the another one is the PUB2223. These are the negative regulator of the plant immunity. As the human has the immunity, plant also has the immunity system in it. So these are the negative. That means, when the pathogen come, they allow the pathogens to colonize and make that. So here we created double mutant, means intentionally we targeted 2 genes, PUB2223, and we created the mutation with them. So I think I'll not go, this is more detailed. So we have done the sequencing to check like the mutation in the individual gene. And then after that, we have done the greenhouse assay. So you have seen all our mutants are very clean. They have not shown any symptom after we challenge it with bacteria, whereas the control plant has completely died. So then, we have published, this is a published paper. After that, we have submitted all the data to the regulators in Kenya, giving them the data on the sequencing, but also all the data required to show that there is no foreign gene integration. So, regulators will not give you like approval like that. They need the evidence to show that, there is no foreign gene. So we submitted they call early consultation form. We submitted all the data to the national regulator in Kenya. And last week, actually, we got the approval where these edited bananas are treated as transgene free non-GM. So they will not go through any regulatory biosafety thing. So it has been classified already. And now we are taking them to the field trial for the BXW resistance, but also to check if they have maintained the yield and everything. Because we want to keep all the traits intact in them. Only the trait we are changing is the susceptibility to resistance. And that's what I think in the morning when you're talking about, some of the varieties which are very good and they have only one issue, we can actually resolve that issue using the gene editing without touching other things in this. So another example in our lab we are doing is like, yam, you see they are 8 to 9 feet tall. So that means the farmers have to do a lot of mechanical work in staking, but then even the harvesting cannot be mechanized. So our breeders actually ask for if we can make this bushy. And this thing has been done in beans, so it has been, there is a lot of information into the literature, but also the whole green revolution was based on the dwarfing of the maize because it avoids the lodging. So that's what we are developing, and we are developing like a bushy yam in our lab using the gene editing. We are also working on the striga tolerance because, as I said, it's a problem in the cereal crops. So, we are working now on the tropical maize using this striga resistance. In cowpea, we are working on aphid because that BT cowpea is commercialized in Nigeria and Ghana, but they can only resist the pod sucking insects, but they don't provide the resistance to aphids or other insects. So we are working on developing the resistance to aphids, use gene editing, but also we are trying to see if the grain size can be increased and the yield can be increased as well. So we are also working on that trait. The another is this Bambara groundnut, which is more like an orphan crop, so not much known. So this is another crop. So this has an issue with adoption because the cooking time is very high. It takes more than 1 hour to cook, and people doesn't have that much time. So with the editing, we are reducing actually the cooking time for this crop. In tomato, there are a lot of varieties, but we are seeing with the climate change, these are tolerance to drought is very poor in many of the varieties, but also there is a bacterial wilt. So we have specific varieties which breeders have given to us. Breeder says that this is the variety, it has all the traits we need, high yielding, everything, but is susceptible to bacterial wilt. So that's the variety. We are fixing it just to add like a bacterial wilt resistance, means knocking out the susceptibility gene there. Or it has all good traits, but is susceptible to drought. So we are working. So these are the three. So these are different varieties we target. We target different varieties based on what actually is a gap there. So that's what the project on tomato is working on. And so I just wanted to show this, that, if you are developing this and if the country has the guideline and if the gene edited product has no foreign genes, so they are treated as non-GM, and then they go the similar path as the breeders use the path, with their hybrids. So that's how it works. But if it has a GMO, if it has the foreign gene, then it has to go to the path of the GMO. So that's the I was mentioning public awareness, and also is still a new technology, so communication and capacity building is very critical. So at IATA we are heavily involved. We train PhD students, so we have a lot of PhD students currently, like about 7 students in the lab from different African countries. We also provide the group training for not only for a few days but longer period, for a few months, or and then we sometimes customize that training based on the need and the request. And we are doing a lot of communication public awareness. Sometimes we feel like seeing is believing. So we also bring people to the lab, and then we show them in the lab things so that they can feel like more comfortable with the things. This is one of the program which IITA is working on. We work with UC Davis under the Plant Breeder Academy, African Plant Breeding Academy CRISPR course. So we run this course every year. And our target is to train 50 to 80 African scientists on gene editing. And then we provide them mentorship. So when they are back into their institution, we provide mentorship so that they can establish. And that's how you see, like, you know, there were a lot of projects which were mentioned, which Nagla was mentioning. Actually, those are our students from the CRISPR course who have gone back, and then now they have a project they are working on. We have trained 32 so far already, and our intention is to run at least 2 more years this course. So if anybody from Egypt is interested, can also apply. We will have the call end of this year for the course starting in January next year. So I feel like the way forward should be like, you know, we only talking will not help, because people needs to see what's there on the ground. So we feel like, you know, there should be very clear way forward for the gene editing. And so gene editing, as you know, enables faster and precise crop improvement, but we need to scale the solutions. And so maybe already what is already in the pipeline, maybe we can give priority to them. Maybe diseases and pests, like, you know, a gene edited banana for the disease resistance. There are also MLN, Striga. These should be the thing which should be scaled so that people really can see the benefit. Expand GED to key agronomic traits like, you know, semi-drought or stress resilience. Strengthen communication to build the public and the stakeholder trust, and build capacity across research, regulation, and deployment. And align innovation with the national priorities. Then only the countries can benefit, and the regulatory framework in that country. So this is actually my team. And then we are partnering with several US universities and also the private sector like Pairwise, because we were initially using CRISPR-Cas9. As many of you know, it has a very complex IP landscape. So now we have a commercial license from Pairwise, Fulcrum, where we have freedom to operate. So, you know, anything we develop, we can take it to commercialization. So we have a website specific for genome editing. So I think if you are interested, you can check on our website. There are a lot of resources on that website. And then we always put the training announcements on the website as well. Dr. Mujib talked about this project already, but maybe I will give like a very briefly, quickly, what the work stream 2, which is on the biotechnology, is about for the biotechnological innovations for sustainable red palm beetle control. So in this work stream 2, we have like, you know, main objective is to develop and implement sustainable biotechnology driven solutions for controlling the red palm beetle, which is a significant threat to the global date palm cultivation. So work stream 2, it talks about mainly like genome editing, the RNA interference, RNAi, and the microbiome base. So these are the 3. And when we talk about the genome editing, so it is the genome editing of the weevil and as well as the date palm. So there are 2 different. So the university in Abu Dhabi they are expert, they are entomologists. So they are one who are working on doing the CRISPR editing of the weevil, where they are making the weevils more like sterile. So like, you know, if you knock out some of the reproductory gene, you can make them sterile. So that's what it. But then in my lab, we are working on the genome editing of date palm, because like knocking out some of the genes which are required for developing the resistance. And so that's what we are working on. Then the similar thing for the RNA interference, we have 2 approaches. One is we want to use like a double-stranded RNA as biopesticide. And so then the issue is like we need very robust delivery system, and then they needs to be quite stable. So we are working on that. Maybe we might use the nanoparticle and other things for the delivery. But then in parallel, we are also working on expressing this double-stranded RNA in the plants itself. So we are working on date palm making the transgenic date palm by expressing this double stranded RNA. And that work is going on also in my lab at IITA. And then ICARDA is focusing on the microbiome-based interventions, where they are exploring interventions to naturally suppress the red palm weevil populations. So I think some of these things I've already talked about. So on the weevils, we are working on genome editing for the knock out the fertility genes. Then there is a double-stranded technologies to silence the essential genes in the red palm. And these both technologies will be then integrated into the pest management. Then on the plant side is gene editing. And we are actually also, the date palm takes longer. So we are using sugarcane as the model. So, in order to with gene, how it will work, we are first testing them in the sugarcane because sugarcane is much shorter. And so we are also using RNAi, as I said, and then both this will require actually the regulatory in framework and the clearance. And then this microbiome, it utilizes the beneficial microorganisms. So ICARDA and Ageri is working on, and they are targeting the gut symbionts as an emerging biological control strategy with low ecological disruption. So any solution which will come out from any of these objectives from the work stream 2, Egypt is one of the beneficiary countries, but we will also seek approvals for testing this in the field here. And then after that, how they can be scale out there. So by this, I would like to thank everyone for your patience for listening to me. Thank you. And happy to take any question.
Thank you, Dr. Lina. Now, Dr. Muhammad will introduce the next speaker, Michael Baum.
So I'm deeply honored to introduce Dr. Michael Baum, the director of the Biodiversity and Crop Improvement Program at ICARDA. Since joining ICARDA in 1992, yeah, I'm talking about you. Dr. Baum has been a distinguished figure for agricultural research in the Middle East and North Africa, leading a diverse team of nearly 100 members. He oversees critical work in different aspects of crop breeding. He has published over 180 scientific articles, book chapters, and conference papers, and mentored a generation of PhD students and postdoctoral fellows. He's a graduate of the University of Göttingen. Dr. Baum has been instrumental in securing over $10 million in research funding and in establishing global partnership with elite institutions like Cornell University and the Australian Centre for Plant Functional Genomics. Beyond his work in the Mediterranean and Africa, Dr. Baum has also served as a guest professor at Guangzhou University in China for several years. Please, sir.
Yeah, good morning again. And actually, some of the introduction is not correct anymore. Actually, officially retired in January 2026. So I'm still supporting ICADA this year as a senior advisor in the office of the DDGI. So, but for the last 16 years, I was leading the breeding program at ICADA, crop improvement programs. So that's why I think I can contribute quite a bit in terms of the biotech application in breeding at ICADA, because as our colleague, the DG from the Field Crops Institute this morning explained, I mean, all these technologies, especially the genome editing, very interesting and very nice. But how do we deliver the products into the field, right? And so one vehicle is actually via the crop breeding program, because institutes like ICADA, together with many of the national programs, regularly develop new varieties. So we are testing advanced germplasm with more than 30 countries in the region. And every year, we release more than 15 new varieties. I mean, it's not ICADA, because the varieties are released through the national system. And but this is happening all of the in the countries in North Africa and West Asia where ICADA operates. So every year, this is one of our KPIs, institutional KPIs. We release more than 15 new varieties. And so over the last 30 years, 40 years, I mean, ICADA was founded on paper in 1975, officially started in Syria in 1978. So over the last 45 to 50 years, ICADA has released over 1,000 varieties. And this is what helps the national programs really to deliver genetic gain in farmers' field. And that's also how we look at genome editing. It's one of the technologies that help to make the breeding more specific, more faster, more accurate, and that's how we want to deliver innovations into farmers' fields. So ICADA is mainly concerned about the dry areas. As you can see, North Africa, West Asia, ICADA has like 3 field operations, one in Morocco, where most of the breeders are based these days, then in Lebanon, in the Bekaa Valley, and in India, in our food legume research platform in Amlaha. So these are the 3 breeding operations, field operations, where we develop new varieties. And every year we provide what we call international nurseries to the 50 or so national programs in that space. So that doesn't work on everything, but on specific crops. And you can, if you want to orient them along the rainfall gradient, we have bread wheat and faba bean. They need a little bit more better conditions, like 400 to 500 millimeters rainfall. Then durum wheat and chickpeas, they do well at 300, 400 millimeters. At a lower scale, we have barley and the lentil at 200 millimeters. So this used to be the traditional crops. And in the last iteration of our strategy, we added crops like cactus, date palm, and forages. Thus also the mandate region of ICADA has been extended. Now we are extensively also working in the GCC countries, in UAE and Saudi Arabia. So that's why we added crops like cactus, date palm, and forages, which are very important crops there. Now, before I come to genome editing, let's talk about breeding modernization, because breeding is a very long process. It used to take 15 to 20 years between the first cross and having a new varieties in the field. And then, of course, nowadays, nobody can wait for the 15 years or 20 years. Everything has to be faster, quicker, more precise. For this purpose, we have heavily invested in our breeding program modernize the breeding. The modernization of breeding, that means there's a pointer. The modernization of breeding no. Let me okay. So part of the breeding is really today everybody talks about speed breeding because you know we need to deliver varieties more faster to breeders, and so that's why you know with glasshouse facilities, and it's possible nowadays to develop 4 to 5 generations per year, and in order to speed up the whole breeding process. But another important component is really high throughput phenotyping, because you know we used to collect the breeders and physiologists and pathologists. They used to collect data in the field with handheld devices. That's a little bit outdated, and there are high throughput possibilities really to do the phenotyping. So, for instance, we have created a heat and drought platform in Morocco, where we specifically can screen plants for heat and drought tolerance, and to select resistant types, and to plug them into the breeding program. So you have devices like these ones, where you can do a full plot phenotyping. There are a lot of cameras in these kind of devices that can do you know, that can do screening of thousands of plots a day and collect millions of data points, and also support the decisions of the breeder to select for certain phenotypes. So genotyping as well as phenotyping, very important that also that we have modern technology, modern tools that can help the breeders to make a faster decision. Then, of course, everything, a lot of things start with a gene bank. So ICADA has an important gene bank for the Middle East, for North Africa, West Asia, which holds more than 150,000 accessions. Roughly speaking, 30,000 accessions of barley, 40,000 accessions of wheat, 70,000 accessions of legumes and dryland forage species. And we use this wide array of germplasm in order to improve our varieties. Because if you want to keep on improving yields and other traits in the varieties, you need to have genetic diversity. If you don't have genetic diversity, you can engineer it, you know. But always the easiest is to look into the land races and the wild relatives if you have traits that can help you to improve the crops. Now that's always the easiest. So since ICADA left Syria in 1992, we have created 2 new gene banks, one in Lebanon, which was inaugurated in 2016, and the second gene bank in Morocco, which was inaugurated in 2022. And this is really, as I explained, I mean, so the land races and the wild relatives, that is what the breeding program makes intensive use of in order to improve the existing varieties. And that germplasm is also provided on demand to any customers, any national program that request a specific germplasm. So Since ICADA left Syria in 2012, we re-established a breeding platform in Morocco. Just to give you a little bit an idea about our operation in Morocco. So in Morocco, we are about the number is varying between 15 and 25 international scientists. We have about 27 national scientists, 50 support staff. We have 100 students, PhD and master and BSc. That's a large part of how we operate. I mean, using masters and PhD students to carry out certain part of the operation. Yeah, that's the varieties released. That's only for Morocco. 20 varieties released since 2012. So we have 6 specified laboratories, office space, the gene bank storage. So we have a budget in Morocco of about $10 million per year. We host a lot of international events, lots of training events. We have the glasshouse space, and we established the speed breeding facility. So that's just a snapshot of the operation in Morocco. So last year, in May 2025, we inaugurated what is called the African Breeding Accelerator. It's a modern speed breeding facility. So we have the capacity to advance about 150,000 plants, and so the you know, and at the moment we don't really need the full capacity of this facility. And we also establishing partnerships with national programs and the private sector who are interested to make use of the facility. Here some more specific information. So it's about 1,000 square meters for 150,000 plants. The LED plants, we have imported them directly from China. They don't use that much electricity because we run them only very early in the morning or late in the afternoon to extend the growing period, and so on. So speed breeding is one of the important components for modern breeding. The other one is genomic selection. So genomic selection, as you can see, started in 2016. Is an international conference on breed in Rabat in Morocco. And here on the screen, you see all the key figures that are important for the development and establishment of genomic selection. At that time, in the middle of the picture, Wayne Powell, he was the CEO of the CG. But for the biometric teams, you see Zakaria Kehel, who is the head of our gene bank, Khaled Al-Shama, who is the biometrician, just at the moment delivering a training course in Syria, Damascus, biometric application in breeding, and Jose Crosser from CIMMYT, who is you know, the most eminent scientist on using genomic selection in breeding program. And so that's another very important component in modern breeding. So genomic selection at ICADA, here's an example of barley. So as I explained, sites in the field are done in Morocco, in Spain, in Tunisia, in Lebanon, as well as in India, 7 sites. So we see genomic assisted sparse multi-environment testing. So that is a special biometrics application they use in the barley program. They reach higher accuracy than before. They can use historical data to predict location instead of environments, and especially allow to recover failed traits, which happens quite often in barley because you know, we had for 6 years drought in Morocco, and it's quite common that traits fail. So we see a genomic selection approach in the barley program. They can even predict the performance of failed trials. So that's very important. When you want to set up, you know, for us, because we are an international organization, but we work with many of the national programs together. we need to ensure that plant breeding is really priority in the national program. So for this purpose, from time to time, as we have done here in Istanbul in December 2022, we assemble all the NAS DGs and really to see if the crop improvement is really the priority. Now here a picture for the legumes, Icada legumes, lentil, chickpeas, fava bean, and grass bean. As you can see, we have asked CDGs, what is a priority? Is lentil a priority? And so, for instance, for Algeria, it was a priority. For Azerbaijan, lentils as well as chickpeas. Ethiopia, all the 4 legumes are priorities, and so on. And that's very important, so that actually, if you because the NAS DGs are important stakeholders, and that we reassure that we do the right thing, and that we are really up to date with our breeding approaches and develop the products that also the national programs really need. So today in that space, all the breeders talk about genetic gain. We need to deliver genetic gain, and the formula for genetic gain is composed of the accuracy, variance, the intensity of the breeding approach divided by the recycling. And as you can see, the recycling time is an important component. If you can reduce the recycling time, then you can also increase the genetic gain. Genetic gain in the breeding program, genetic gain in farmers' feed. That is really what we need to deliver. And here some idea about our legume species. So in the speed breeding, the accelerated breeding, so between the vegetative phase and maturity, the legumes need about 60 to 70 days. So in 60 to 70 days, we can have a generation, and so very similar to the cereals. And so 70 days for one generation, that means we can add up to 5 generations here. That's really modern plant breeding that you are much faster in developing your products. And so we have calculated, we are annually calculating the genetic gain in the breeding programs. We are at about 2% delivery in the CG. The breeding programs are supported by the Gates Foundation. The Gates Foundation require a minimum of a genetic gain of 1.5%, and so we are above this one. And but this is an average figure. There are certain market segments where we have much higher rates, even 4%, 5%, and others which we are much less. And but as an average, for all the breeding programs that we operate, we are at about 2% genetic gain per year. Now, if we talk about what are the best technologies, the appropriate technologies, so for instance, gene editing versus accelerated breeding. So because certain targets you can achieve with accelerated breeding. For other traits, you need really genome editing in order to achieve your goal. So to answer the question, what genome editing targets? If we have well-known alleles, and here the genome sequencing and the plant genome approaches are very important, because here we do a lot of sequencing. We have a lot of sequence information, and so to identify the genes that we want to modify in the breeding, as well as with the genome editing approaches. And how is not all the varieties are amenable to genome editing. Genome editing effective today for knockouts. So that a lot of the cases where genome editing is applied is really to knock out a certain gene or to modify a certain gene, and rather than adding a lot of sequences or deleting. The bottleneck is always that in most cases, we need still go through a color space for multiplication. But here we really can build on the early generations of GMOs, because we have developed transformation protocols for many of our crops. And even today with the genome editing, we still need to have the transformation protocols. So we can actually recycle, I mean, the work that we have done before. And so as far as ICARA mandated crops are concerned, so we have the editing protocols for in lentils and chickpeas, as well as in wheat and barley, for that we can use for the genome editing. Now, for an international center, so for major crops like wheat and barley, we really seek the collaboration with more advanced research institutes. Like, for instance, here in Bali, we work with Professor Maria Koch from the Institute of Plant Genetics in Germany, in Düsseldorf. And because the cereals, it's a very highly competitive space. And so here we are looking for a number of genes that are related to rust resistance in barley, and with the John Innes Institute in UK for rust resistance in wheat. Yeah, so these are some of the targets for barley. And so that's, that's the status of ongoing work in barley. But then as an international center, I think we really have a role to play in the legume species. That's where we could be competitive. And so ICARA works on the 4 legume species: lentil, chickpea, faba bean, and laturus, as well as forage legumes. And so here we have a few very important targets for genome editing, such as Ascochyta blight resistant is a major disease in chickpeas as well as in lentil. Here we could knock out a few susceptibility genes and fine-tuning resistant pathways. Herbicide tolerance is very important for the legumes, especially if you want to enable conservation agriculture. The plant architecture, many of the legumes are determined, and so you can play with determination or determination. Lodging resistant also. Seed quality is important. Anti-nutritional factors in faba bean, for instance, or the beta ODAP, which is a neurotoxin in laturus. The flowering control, the matching crops with shortening seasons. And then, of course, abiotic stress, salt and drought tolerance in chickpeas and lentils. Now, as I told you from the earlier work on GMOs, we have developed the protocols, the regeneration protocols for chickpea. And here are 2 of those employed. It's the Agrobacterium-mediated transformation via mature embryo explants, as well as the Agrobacterium rhizogonous-mediated hairy root transformation via cut dipping. So these are the protocols being used. And you will see that in the coming days in the practical session. And so we have a number of examples of chickpea plants in with different construct and variant in various different development stages. So one target here that is being handled at Ageri and that is funded by STDF, I mean the Egyptian funding agency, is improving drought tolerance in chickpeas by genome editing. And here especially 2 different genes being targeted. One is the ERA1 mutant. The aim is here to overexpress to ABA. The phenotype is more closed stomata phenotype, and this leads to improved drought tolerance. The other gene is the SAL1 gene, inositol polyphosphate 1-phosphatase. that also targets drought stress tolerant because the gene is involved in stress signaling and redox homeostasis, and the modification enhances tolerance to abiotic stress. So these are the two genes the colleagues at Ageri currently working on. As I said, Lativirus is another crop for ICADA, which is of regional importance in the highlands of Eastern Africa, like Ethiopia, as well as in India, Nepal, and Bangladesh. Grass bee has one problem, it's beta odor. Beta odor is a neurotoxin in the sea. And for the last 30 years, the breeding programs have tried to reduce the beta odor concentration in Lativirus. But now with the genome editing methodology, it's really possible also to modify, the genes that are responsible for beta odor. And in a project with between ICADA, Queensland University of Technology, and the John Innes Center, mutants, genome edited plants have been developed that have zero concentration of beta odor. And as I said, it's possible to modify some of the pathways in the plant that can be very useful for breeding application. A lot, as I said before, we in the 90s and 2000s we developed some of the protocols for the genetic transformation at ICADA. This was our facility in Aleppo, Syria, till 2012. Then, unfortunately, we had to leave and shift whatever we could rescue to Ageri, where we're operating today. We're very grateful that we found a new home in Egypt and in Ageri. And so where we can really use what we have learned over time for the benefit of the ICADA mandated crops, chickpea, lentil, barley, and wheat. And so that's in a nutshell how we think that we can deliver, improved varieties, and then also plugging in genome edited plants into the breeding programs to deliver for the benefit of the farmers in C1. Thank you very much.